Right, welcome back. The first South African and African COVID-19 vaccine trial is currently being announced. It's being hosted by Wits University, and I understand that Professor Helen Rees, she uses a number of hats. Professor Helen Rees, the chair of the African Regional Immunization Technical Advisory Group of the World Health Organization, chair of the Program and Policy Committee at Gavi, the Vaccine Alliance, the chair of the South African Products Regulatory Authority, among others. Let's listen in.
Or I know somebody who had that disease and I want to protect. I don't want to see that happening again. There is a true sense of altruism from many people who participate in trials. And because this is a global pandemic, there needs to be that opportunity for many people around the world to be able to express that solidarity and that global altruism. So that's the second reason. But the third reason is that if we participate in trials, there is quite a strong moral obligation for us to be able to say, we've helped develop what we hope will be a successful vaccine. Therefore, we want to ensure that the people in the country where it was developed and the region in which it was developed have access to those vaccines. Unfortunately, what we're seeing at the moment is what we're calling vaccine nationalism, that in fact vaccine production is being ring-fenced for particular countries. And obviously the richer countries and the richer areas are trying to say, my country first and all my citizens first. So there's a very, very important global dialogue now going on about vaccine access, which says, actually, if we develop vaccines, these need to be available to all citizens of the world. There might and will have to be prioritization because the amount of vaccine available to begin with will be limited. And we might have to think about groups such as healthcare workers first. But what we are arguing is that it cannot be my country and my country only first. So what started off as a conversation about pathogen X has become a massive global discussion about access. And one of the last things I'll just mention in this regard is the impact of false news, of anti-vax movements, and of a very unfortunate incident that occurred a few months ago where two French doctors had a dialogue where they talked about vaccine trials in Africa, and it was interpreted as being a very oppressive and exploitative approach to participation in vaccine trials. And it has caused a lot of discussion and a lot of concern in the African region. But we have to counter that with the arguments that say, actually, we want to be in trials. We want to participate. We want to be part of this scientific endeavor. Minister Zweli Mkhize has been part, as you know, we're leading the African Union at the moment, has already entered into that dialogue on behalf of the African Union about access for the African region and is totally supportive of South Africa's participation in trials. Thank you.
Thank you for Professor Rees, and also thank you for giving us the background into the story, and most importantly, for clarifying the issue that raised its ugly head a few months ago. Now, I'll hand over to Professor Gray, the President and Chief Executive Officer of the South African Medical Research Council and a research professor in the School of Clinical Medicine at the University of the Witwatersrand. Over to you, Prof. Gray.
Thank you. DG Sandile Buthelezi, Zeblon Vilakazi, Johnny Mashlangu, other colleagues, all protocol observed. It's wonderful to be here today to witness the announcement of the first coronavirus vaccine to be studied in Africa and indeed in South Africa. So the South African Medical Research Council is a major funder of this program. And for those of you who don't know, our mission is to fund and conduct research that changes the lives of South Africans. So you can see why this is a very important study for us. Before the epidemic started in South Africa, a group of scientists, together with members of the Department of Health and the Department of Science and Innovation, got together and developed a research strategy for South Africa to try and see how we could make local innovation to combat the COVID epidemic in South Africa. And we were able to mobilize resources and get funding from the Department of Science and Innovation and release other funding from the MRC to conduct to start to fund a whole lot of research in South Africa. And this research started off with surveillance, trying to understand how the epidemic was unfolding in South Africa. It also entailed investment in finding local solutions towards testing. We also are collaborating with other funders to fund clinical interventions and therapeutic interventions for COVID infection, as well as understanding the immunology of the COVID epidemic in South Africa. What's very important as well is the issue around prevention, and we became very interested in funding work on vaccine design and development. And we looked at both local vaccine design and development as well as collaborating with international groups. And so this is the reason why we have collaborated with the Gates Foundation to fund this very important research that looks at both HIV-infected and uninfected people and tries to understand immunology and also answer the issues of preliminary efficacy. So this is very important. And this is one of 3 vaccine trials that will start this year. Very soon on that, we hope the AD26 J&J program will start, as well as an AD5 program that we're collaborating with people from Los Angeles. And so we are very proud and very excited to see this program work. And we look forward to seeing Shabir expand this to the rest of South Africa. And hopefully the results will be very encouraging. So it's very great to be here. And thank you very much for allowing us to participate.
Thank you, Professor Gray. And colleagues, just a reminder that a press pack was emailed to all of you a few moments ago. So, following Professor Madhi's presentation. So now with Professor Johnny Mahlangu having joined us, I'd like to hand over to him, who is the chair of the South African Medical Research Council and the head of the School of Pathology at Wits University.
Sorry, Professor Vilakazi, he's still not able to talk to us. He's in the meeting, but his microphone doesn't want to work. So I think if we can just carry on, we're just trying to get him to be able to connect to us.
All right. So then I'll hand over. Thank you, Craig. I'll hand over to Professor Jeffrey Mphahlele, the Vice President of the South African Medical Research Council. Over to you, Jeffrey.
Thank you, Program Director. And good morning, DG, Dr. Sandile Putelezi and colleagues. I think it's quite fitting that we need to congratulate Professor Shabir Madhi for this groundbreaking and world-class research, because from the perspective of a Health Science Council, as the South African Medical Research Council, clinical research certainly offers great opportunity to improve and advance the quality of health. So a new pathogen like SARS-CoV-2 really requires rapid and responsive research with a view to optimizing development of new health solutions, as you have already heard. So the need for a vaccine obviously cannot be overemphasized, and only vaccines are the only proven public health biomedical interventions to control, eliminate, and also eradicate infectious diseases. We know that it is through the vaccines that we have been able to control dreadful and fatal infectious diseases like measles, whooping cough, and hepatitis B. And it's actually through vaccines that we have been able to eliminate other infectious diseases like tetanus. And also, just to add that it's through vaccination that we have been able to eradicate smallpox. And the world now is at the cusp of eradicating another infectious disease, polio, which was crippling and a deadly infectious disease as well. So we really know the importance of vaccines, and the R&D in vaccine development is necessary and is something that we need to support at this stage, because it's only through vaccination that we can contain this pandemic, which is due to COVID-19. So we are hopeful about this trial that is going to start tomorrow. And we think that should a vaccine be developed, the first thing is that we wish for is that the vaccine would be highly immunogenic in the majority of recipients. We know that this might be difficult in other age groups, like you heard from Professor Shabir Madhi in the old age group. But we are hopeful that the vaccine will confer long-lasting or even lifetime immunity or protection in the majority of recipients in all age groups. Because if that is the case, then we know this COVID-19 vaccine will form the backbone of national immunization programs in many parts of the world. And we need that kind of a vaccine so that we can accelerate the control of COVID-19. But not only the vaccine that will become the backbone of public health vaccination programs, but the vaccine that will offer an excellent example in terms of a life course immunization from childhood to elderly, because we know that this COVID-19 is affecting all age groups, and a vaccine that is under development now is an excellent candidate for a life course immunization. So with these few words, we like to really congratulate and support Wits University for this endeavor, and we hope that a vaccine will be found. Thank you very much, Program Director.
Thank you. Professor Mphahlele, for those kind and encouraging words. And now we'll hand over to Professor Dr. Sandile Buthelezi, who will give us a final word, who is the Director General in the National Department of Health. This will be followed by the Q&A session. And I see a few questions have already been responded to by the panel, but we will now leave the platform open after Dr. Buthelezi has spoken. Over to you, Dr. Buthelezi. Dr. Buthelezi, can you unmute, please?
You're mute.
Hey, one before technology. Thank you very much, Professor Vilakazi, and a good morning to the esteemed professors in the panel and members of the media. As a Department of Health, we are very happy, and we actually would like to congratulate Professor Madhi for this groundbreaking launch of the vaccine here in South Africa. But secondly, we are proud that we are associated with Professor Madhi as a member of the Ministerial Advisory Committee on COVID-19, together with the other professors on the call here today. But thirdly, also, we are very proud that South Africa again has been placed in the center stage of leading in science. And as a department, we are always saying that South Africa must always lead from the front. There would have been no better time than today to actually launch this vaccine trial, as the country has reached a landmark, more than 100,000 infections, which we recorded last night. And we've also reached high levels of deaths in the country. And we really believe that it's a way to go. Of course, we currently now only depend mainly on non-pharmaceutical interventions. So if there's any way that we can fast track our road to getting the vaccine, that will be the long-lasting solution that we'll have as a country. I must say that As a country, we are actually...
Yes.
Yes, as a country, we are currently on stage 5 to 8 stages in terms of the management of the epidemic. And we're really focusing mainly now on hotspots. We've got about 29 in the country in the 4 high burden provinces. And we are really having targeted interventions, health facility readiness, and ensuring that there's proper clinical management of patients that present to the facilities. So all this work, of course, we really hope that once we get towards prevention through the vaccine, and we pray that this vaccine development will actually yield positive results in terms of immunogenicity. And we thank the team, especially Professor Madhi, MRC, Wits University, and all other collaborating institutions, and we pledge our support as a Department of Health. Thank you very much.
Thank you. Thank you, Dr. Buthelezi. And we are delighted to note that the university and other universities and research entities in the country will have the support at the national policy level through the department and obviously the ministry and the government at large. And your words are very encouraging indeed. So, colleagues, this brings us to the final session of the Q&A. So what will happen is that some questions, I'm aware, have been already fielded and responded to by some of the panelists. So they will come to the Q&A channel. And, please, again, state your name and organization. Who would you like to respond to your questions? And, of course, please unmute yourself when asked to do so. So most of the panelists will be available for interviews after the session. Should you wish to set up follow-up interview, please, you can email them to vets.news, NEWS, @vets.ac.za. And so this starts our 30-minute Q&A session. And I see, if you help me here, bear with me for a second, that there are questions that have come through. The first one comes from Graham Hoskin from Sunday Times, who asks, please, panelists, note this. How were the people taking part in the trial selected? First, about the selection of the individuals taking part in the trial. The second part of the question is, what kind of questions were they asked? And the last one is on the gender split between men and women of the initial cohort. So I suppose you'll go through question by question. So who is the respondent? Will it be Professor Madhi?
So thanks, Zebra.
Shabir?
What we've done is that we've basically just sort of sent out messages in Soweto in terms of, at least that's from at my site. Like I said, currently in Gauteng, there are 3 sites that will be involved. And we've been sending out messages to the community in terms of people that might be interested in participating. And to give you as an example, last week, when we sent out our initial message, we had close on to 1,000 people that arrived at our clinic in a space of 3 days, wanting to know more about the study as well as wanting to participate. So once an individual comes to the site and indicates an interest to participate, what happens is that they go through a process known as informed consenting, where the staff that are involved in the study will basically engage with the individual in terms of understanding what is their interest in terms of wanting to participate, as well as explaining to them what participation in the study would actually entail. After that process, what next happens is that the participants are actually provided what we call an informed consent form, and they actually go back home and they've got the luxury of being able to read it at their own convenience. They are then brought back, and that has started as from Monday. They are then brought back to basically, if they are interested, they would come back, and then they would engage with the investigators again in terms of whether they're still interested or not. And if they are interested, the investigators would then ask them to complete a questionnaire, which basically involves trying to get an understanding as to how well they actually comprehend what it means to participate in a study, including issues around risk and in terms of what is expected of them. So it's sort of a 10 questions that are asked, which does a sort of a macro assessment in terms of their knowledge. And it is only if they score 8 out of the 10 that they are actually allowed to participate in the study. If they do not score 8 out of 10, we try to go through the process again to heighten their level of understanding of the study. In terms of males and females, there isn't any sort of predilection for wanting to enroll one or the other group. We're wanting there to be some sort of a balance between men and women that are eventually participating in the study so that the results can be more generalizable.
Thank you, Prof. Madhi. The second one is just a comment from the colleague at Wits, which is supportive. Thank you, Robin. There's a question from Robinson Ngola from Power FM, who asked about the cost of the study. And again, the second part is, when do we expect it to have it rolled out if it's approved? So the costs and the timelines of the rollout process.
Yeah, so in terms of the costs of the South African study, usually it costs a few $1 million to be able to do this sort of studies. The total cost of the South African study is roughly about 150 million rand. That would be the cost of the South African study. And that is really a study which is done at a relatively low cost compared to what it usually entails to do this sort of intensive studies. In terms of when the vaccine will be rolled out, I think the reality is that our best case scenario is that we would be able to have an answer for this particular vaccine by the end of this year. That is our best case scenario. It might take longer depending on how quickly we are through outcome cases. But even after that has been completed, it doesn't mean that the vaccine becomes available immediately. Because what needs to then happen is that there needs to be manufacturing plants that are set up, which is currently being done for this vaccine, both in Europe, in the United States, as well as in India. And then the product that comes out from those manufacturing facilities needs to be evaluated in terms of ensuring that they're sort of similar to the product that was evaluated. So the best case scenario in terms of when this vaccine might become available will probably be in the third quarter of 2021.
Okay.
Can I just add something to that? In terms of vaccine availability, I think that this is a really critical thing to understand, because even if we have successful vaccines through clinical trials, we will not have enough vaccine by the end of 2021 for the whole world. We're talking billions and billions of doses. So we are going to have to look, as I say, at prioritization. So there is an entire global effort at the moment, which is looking at how do we prioritize, who do we prioritize, how do we prioritize? And that will depend on the kind of vaccine that is successful, because different vaccines have different characteristics. But that is where this global debate about access to vaccines becomes very important. And because it's the media, I would really, really love it if the media takes up this call. We had the same issue with access to antiretrovirals, and now we're seeing a replay potentially with access to a successful vaccine. So I think that this is a drum we must really hit very hard in our region, because we will be the region that will be left behind otherwise. This is what happened with the pandemic flu vaccine. Africa got very little vaccine and we got it months too late. And we cannot allow that to happen again. So I would really encourage media to take an active interest in this in terms of this global debate.
Can I add, Zeblon?
Yes.
Can I add? I just want to say that that's also why it's important for us we hope that not only one vaccine will be successful. We need 5 or 6 vaccines to be successful. And so to make sure that we can scale up to the billions of doses we need at a global level. And also why it's important for South Africa to be involved in not only one, but many efficacy studies, because if we are involved in the efficacy studies, we know exactly how we managed it in South Africa, and the regulatory authorities will have experience with the vaccine, and will make it easier for it to license. And so having local experience with an experimental vaccine is critical to the endeavor for licensure. And also, it's also critical because if we are involved in efficacy studies, it gives us first in the queue for access afterwards as well.
Thank you, Glenda and Helen, for the follow-up question, for the follow-up responses. Now, on funding again, I think one came out from Liz Cummins from Independent Media, the Mercury newspaper, regarding funding. So I think I want to just maybe follow up on that. She asked, how much funding have the two funders made available for the trial? I mean, are members of the panel able to respond to that?
So the South African Medical Research Council is funding the HIV-infected component of the CHIMP-AD program, and it's costing us around 10 million rand. And that's given the fact that we only have around, we've managed to secure about 100 million rand to do research in the COVID vaccine, COVID epidemic. You can see that this is a huge portion of our funding, but we remain committed to working with other entities to raise more money to fund further vaccine trials. And if the CHIMP-AD program is found to be effective, we will work with the NDOH and the Department of Science and Innovation to raise more money to fund this further.
Thank you. Vinicius Assis from Brazil asked a very interesting question, which most of the panel members can take, is in terms of comparison between how South Africa is managing this pandemic and, of course, with his home country, Brazil. And therefore, what are the impressions of the panel members in terms of how do the two countries respond to the pandemic? I mean, that's a tough one. I don't know who's going to answer that.
I'll follow Shabir.
So I'm happy to speak.
Shabir?
I think Brazil, obviously Brazil is a huge country, and each of the different states in Brazil have had varying responses. But at a leadership level, certainly the approach of the president in Brazil has been very different from the key leader. The president of South Africa has been probably diametrically opposed to what has happened in Brazil in terms of the manner in which the Prime Minister has basically gone about responding. So South Africa has been much more aggressive than Brazil in terms of trying to slow the rate of transmission of the virus. They've been wanting to be much more enforceful in terms of getting people to adhere to the non-pharmaceutical interventions. But these are both complex societies, both Brazil as well as South Africa. And I don't think it's a correct thing to make comparison between countries. Each country has got its unique challenges, but South Africa, that being said, I think South Africa and Brazil have got a number of similarities. It's an example in terms of inequity within those sort of societies, as well as issues around access to health care. But it's unfortunate that Brazil is seems to be ahead of the curve compared to where we are in South Africa, and we can only hope that what we experience in South Africa in the next month doesn't mirror what is happening in Brazil at this point in time.
Yeah, it's like Brazil is a low middle income country, and we will have similar epidemics because of our Gini coefficient and our inequity in terms of health access. Also, our overcrowding and our poverty and issues around lack of employment. And I guess what is different to South Africa, if we look at Brazil, is the issues around hydrochloroquine, some of the issues around, first of all, the importance of establishing evidence of interventions before rolling out or endorsing them. So I think those are important issues. I think that Brazil is struggling, from what I can see from the media and in the newspapers, is that there's a huge struggle. But what's very exciting about Brazil is that they are involved in 3 or 4 vaccine endeavors, and I think that's going to be very exciting to see how Brazil responds and how they are able to roll out vaccine endeavors, and to see whether it works and work with South Africa in that setting. And so, as Shabir says, no epidemic is the same, and it's very hard to draw parallels because we all have our own issues and we all have our own struggles, and we also have other health and economic issues that are quite different to different parts of the world.
Could I add just one?
Thank you.
Could I add one more comment to that? And that is that, and something else that we share with Brazil is a long history of doing HIV research. And one of the things that's very noticeable worldwide is that, and particularly in South Africa, is that quite a lot of the experience that we've gained through doing HIV research and TB research, we're now able to turn to COVID-19. I mean, Shabir described very well extensive experience with vaccine development as well. But that is something that we do share with Brazil, and it's given us exceptional scientists, which I think we're lucky to have, and who are now turning their minds to a new challenge.
Thank you. Just a follow-up question on that, Helen, or maybe to Shabir, I don't know, between the two of you, and Glenda as well. It's also a follow-up question by Graham Hoskin linked to the HIV link is as following. Graham Hoskin asks, also from the Sunday Times, how many HIV positive people are taking part? I think Shabir did answer that, but can you just elaborate?
Yeah, so for HIV group, it's basically only 50 participants, half of whom will receive the vaccine and half that will receive the placebo. And what we're really trying to do there is just to see how well they respond to the vaccine in terms of the immune responses. And using the data from the HIV negative group, where we're looking at immune responses as well as whether the vaccine works or not, we would sort of be able to use the immune responses readout from the HIV infected individuals in terms of getting an estimate as to how well the vaccine would work in that group compared to the HIV negative group. So even though we're not looking for efficacy in the HIV positive group, using the immunogenicity data, we can sort of do bridging in terms of understanding how well the vaccine would work in that group. So we won't need to repeat a full study if the immunogenicity is comparable between people with and without HIV, and the vaccine works in the HIV negative group.
Okay. Any follow-up responses?
Just to say that in the efficacy studies, one will not discriminate on HIV status. And so when we're looking at the design of the phase 3 studies, we will take all comers and HIV infected and uninfected. We will obviously will look to see whether there is a difference in efficacy and whether HIV affects the shedding or the protection in any way. So I think that it's very important, and we will make sure that in certainly the phase 3 that I'm involved in, is that we will take all comers irrespective of HIV status or TB status.
Can I just make a personal extension of the question, actually? I mean, as take advantage of the position of the chair to ask whether does the same apply to TB as well?
So in the current study, if someone has got past TB, they would be included. If they're on stable treatment, they would be included. If they are newly diagnosed case of TB, we need to prioritize them getting treated for TB rather than entering into the study. So they will be excluded if they got active TB that hasn't been on long-term medication or newly diagnosed. Priority for those individuals.
You have to stabilize people who are ill first, Zebulon, and so they would need to be stabilized before they would be ready for enrolling into a trial.
Thank you, Glenda. Thank you, Shabir. Now, there's another question from, it's the first question actually, from Tabil Mbele, from SABC Radio News, who asks, under what circumstances would you, suspend or cancel this trial? Under what circumstances would you suspend or cancel this trial?
Yeah, so I think that's a really important question. And as I alluded to in the presentation, throughout the study, including the study that's been done in the United Kingdom and in Brazil, in real time, there's a independent data safety monitoring committee that reviews whatever is happening in the study. And if there's any concern on their part, they would be able to actually work out whether the person is actually receiving the vaccine or placebo. And if there's any suggestion that the vaccine is doing any harm, irrespective of how many people have been enrolled into the study would be terminated immediately. So the Data Safety Monitoring Committee has actually got a prerogative to be able to tell investigators to stop the study immediately. So that's the one case scenario. The second case scenario is if we find a huge number of people that are infected over a short period of time, and we're able to then analyze to whether the vaccine works or not. So if we reach 42 cases very rapidly, without having completely enrolled the 2,000 individuals as an example, we would then already be able to see whether the vaccine works or not. And at that point, we will stop enrolling further into the study. So the study can be prematurely terminated either because there's a safety issue, which the Data Safety Monitoring Committee picks up on, or alternatively, if we find that we're getting large number of cases over a short period of time, which allows us to be able to do an assessment as to whether the vaccine works or not.
Okay. Thank you, Shabir. Then we have a question from Catherine Child from Business Day, who wants us to clarify how many people in the UK this vaccine has already been tested for in terms of safety.
Yeah. So right now, there's 7,000 participants in the UK that have already been enrolled into their studies, which includes evaluation of safety and immunogenicity, and they've been followed up for efficacy. And that 7,000 will increase to 10,000 by the end of July. So the UK has already enrolled 7,000 participants in their study.
All right.
Zebulon, just to add that, and in the US, over 10,000 people will be enrolled over the next couple of weeks as well. So there's going to be massive expansion and evaluation of this vaccine all over the world.
Just a follow-up question that maybe, I'll actually ask Helen to field that one, or John, or Jeffrey, is from Catherine Child, a follow-up to the earlier one from Business Day. She asked, why do we expect some vaccines to work when, for example, there's no TB vaccine after years of study? There's no TB vaccine after years of study.
Well, we have BCG, and we're looking at some others. So, but it's a fair question. I think the first thing to say is that every pathogen, in fact, if Johnny could speak, he's the one that should answer this, and Jeffrey, actually. But every pathogen is completely different. Look at the years of study for HIV. And some pathogens have proved to be easier to form to develop vaccines for than others. So that's the first thing to say. The second thing that I think is somewhat encouraging is that we have got a backdrop to the development of these vaccines, which comes from MERS and the original SARS outbreak. So vaccines were in development for both MERS, in fact, currently are in development for MERS, and were in development for SARS. So we had vaccines that we'd already seen had very strong signals, both from animal and preclinical data, but also in some cases from clinical data, that these would be effective. But of course, SARS went away. But MERS vaccines are still being developed, and these are also coronaviruses. So that's the second thing that gives us optimism. Is a similar in this class of virus, it looks like we can do this. The third thing is, and it goes to an earlier question we didn't touch on, is why select some vaccines and not others? And of course, it depends who's at the front of the queue. I mean, that's part of it. But also, we do get a sense of how vaccines might work by looking at the preclinical data and the animal model data. And if that looks hopeful, then there's good reason to be optimistic that we will be able to find vaccines that work. I think my last point on this, and Shabir showed it very clearly, is that we're using not only established vaccine technologies here, but we're using completely novel vaccine technologies, which might in fact give us a sort of a head in this race by using things that we think might be very immunogenic and really cause a very strong response in the human immune system. So, but we'll have to wait and see. And let's hope we're not like HIV in the end of this.
Thank you. I see we've got Johnny. Oh, Johnny's back. Johnny and Jeffrey, can you please from your
apologies,
technology point of
Chair.
view?
I had some technical difficulties, but I agree completely with Helen's reasons why we should continue to try. And the one reason, perhaps in my view, that is also important is that this particular condition currently does not have treatment or cure. Prevention is our approach to managing this condition, and developing a vaccine is probably the best approach that we can do in the absence of a cure.
Thank you, Johnny. Jeff?
Yes, thank you very much, Program Director. Maybe I can just add to what my colleagues have already indicated. I think from my side, the fact that we have not seen reinfections from people who have recovered from this disease is a sign that there is some form of protection if you get infected. So we have seen that globally, there are a number of people who have been exposed to the virus, and they, cleared the virus, and they developed some form of protection. We just don't know how long this protection is going to be, whether it's going to be a year or maybe a couple of years or for a lifetime. We don't know at this stage, but it looks like there is a sign of protection. So that's the first thing. The second thing is that for vaccine development, you definitely need to identify what we call correlates of protection. And in this case, with COVID-19 virus, we know from many animal studies that the spike proteins that are on the surface of the of the virus, they seem to induce protective or neutralizing antibodies. So that is important, and we have also seen that for a number of vaccine candidates, if you use these spike proteins, they can induce some form of protection. And then the last thing is that we seem to be lucky with this virus because it's genetically stable. So we don't have a lot of mutations. And because of that, whatever vaccine that is developed, it should be able to work in different parts of the world. Thank you.
Thank you, Jeff. I just have another follow-up question from Graham Hoskin. He wanted to understand from Helen regarding novel technologies that Prof. Rees, you referred to. Can you just give a quick answer to that? Because I know that you're under a bit of pressure of time now.
Yes, and
Helen?
You'll have to excuse me because I'll have to leave this. I think Shabir showed it very nicely in one of his slides, that there are a range of technologies that are being looked at in terms of the vaccine construct. And some of them, as he described, are things that are tried and tested and that we use in current vaccines. And others are really very novel and we haven't got vaccines yet, but they have different ways of working. And the ways of working would influence things like how much an immune response they evoke, whether you need one dose or you need 2 doses, and what kind of side effects you could anticipate in advance with different constructs of a vaccine. But I don't know if Shabir, you want to come back and just revisit.
I can also add, so the ad-based platform has been very useful. So both for cancer, as I think Johnny also mentioned, as well as for Zika and for Ebola. And so the platform, so the Ad26, Ad5, and Chimp Ad platforms have been critical for other vaccine programs. So to understand the immunogenicity and then to use specific antigens, as Jeffrey said, using the spike protein into the Chimp Ad platform is a modern way of approaching emerging pathogens, and there's been good success with Zika with the Ad26 platform using that approach as well. Now with the Chimp Ad as well. So maybe you can talk about the role of adeno-associated platforms for both cancer and new viral infections like Zika and Ebola re-emerging, and now with the coronavirus. Shabir?
Yeah. So thanks, Glenda. So I agree with what Glenda said. I think the important thing is that even though there aren't vaccines that are licensed, which are sort of DNA or RNA vaccines or the viral vector vaccines, which is a new technology that we're referring to, this technology has been used for other purposes, which I sort of alluded to in terms of, as an example, developing vaccines against cancer or trying to correct for genetic abnormalities. So this technology has been used for other purposes. And one of the major advantages of these technologies is that, firstly, in terms of scaling up production, there's much greater potential to scale up the production of these nucleic acid sort of vaccines or viral vector vaccines. The chimp vaccine that we're looking at, in fact, this particular vector has already been used to develop a vaccine against SARS virus and a MERS virus. So that was in 2002 and 2012. So this technology was already used to develop vaccines against those organisms, but fortunately, those sort of outbreaks burnt out, and there wasn't any need for further development. So like Glenda mentioned, there's a number of vaccine constructs, as you can see on this slide, more than 25 just at this time. That number has probably increased to 50. In terms of the number of people that are looking at viral vector vaccines. But I think importantly is that this technology has been used previously for other purposes, and it is licensed for other purposes.
Johnny, maybe you want to comment about the role of this in genetic... Yeah. Johnny might want to comment about the role of these in genetic abnormalities and your work that you're doing.
I'll be very brief, Glenda. I think we must just keep in mind that whilst we all strive for preventative vaccines, there may well be other influences, including the genetics that might allow some individuals to respond better to vaccines than others. I think that is an obvious thing that we always keep in mind. But we don't use that as an exclusion criteria for individuals to participate in vaccine trials such as this one.
Thank you very much, colleagues. I note that we are slightly running out of time, so I'll just take maybe a just quick follow-up from Catherine, cluster it with the other one as well, regarding Catherine Child asked, is trial wanting 100% protection against the virus, right? Low or higher? What is the efficacy of that you expect? And it's linked to one by Mia Malan from Peggy Sisa. So I want to just cluster them. What minimum level of care will be provided to participants? That's an easy one to respond to. If colleagues, one of you can just do them as a cluster, and then you can just do two more questions because we're running out of time. Thank you.
So, Zeppelin, I'll take that. So, there's no vaccine that's ever going to have 100% efficacy. There is no vaccine that has 100% efficacy. The way the study is designed is that we're trying to establish whether there is at least 60% efficacy. So we got enough number of individuals participating that allows us to assess whether there is at least 60% efficacy. And I think we shouldn't get sort of hooked onto this issue as to a specific point estimate, because what's more important than efficacy is what we call the number of people that need to be vaccinated to prevent one illness. So there's another measure which is much more important from a public health perspective, in that you can have a vaccine that's only 50% efficacious, but it can prevent more people from becoming sick compared to having a vaccine that's 90% efficacious, but for a disease that is very rare. But the short answer is that we're trying to see whether there is at least 60% protection against COVID-19. It might be higher, it might be lower. If it is lower, our study wouldn't have enough what we call power to be able to conclusively say that the vaccine works or not. In terms of protection to study participants, all of the study participants are obviously counseled in terms of the importance of non-pharmaceutical interventions. The study participants are provided non-surgical face masks, they provided sanitizers, and that is as far as we can go in terms of trying to get people to understand and to get them to minimize their risk of becoming infected. So even though the study is very dependent on people getting infected, which is going to be an unfortunate reality, at the same time, we're making all efforts within our disposal to get people to understand the importance of non-pharmaceutical interventions and providing them with whatever tools we have available to us to reduce their risk of becoming infected.
Thank you, Shabir. Just a nice follow-up question on that, and then before we start closing, with two more quick questions, is about the efficacy and genetic, population genetics, right? So what works for the UK or Brazil will not show efficacy among our local populations, because the vaccine was developed in the UK. So in terms of the genetic response factor, what are the variables do you have in mind, Prof. Madhi?
Yeah, so absolutely important question. And that's the reason why it's absolutely essential to actually evaluate these vaccines in different settings. Because we do know with COVID-19 that there is possibly a genetic predisposition in terms of who is developing more severe disease. As an example, people with blood group A seem to be more susceptible to developing severe disease. As part of the studies, both in Brazil, in South Africa, as well as in the UK, we are actually doing some genetic analysis of the participants. And at the end of the day, we'll be able to combine the results from all three studies together with the genetic information that we've been able to accumulate to see to what extent the genetics of an individual might lend itself to increased susceptibility for COVID-19, as well as what impact it might actually have in terms of the vaccine efficacy.
Thank you. Colleagues, just before I close with the last two questions, and then I'd just like to also acknowledge the Dean of the Faculty of Health Sciences. As you know, most of the researchers from VETS are within that faculty. Professor Martin Vela, who's here, is basically under his guidance as the Dean of the faculty that the appropriate resources were made available and the academic environment allowed for conditions for such excellent work to be done. Thank you, Martin. I have just one question that is linked to the first one about the Tanya Faber. I think Helen did answer it, but the damage about fake news that came out from about Bill Gates and so-called unethical trials in Africa. I mean, there's fake news, but I think there's been damage. And the other one that is linked to that, I think they're both the last part of the class of last questions, is there's obviously a global debate. This question comes from Freda Ho from Spotlight. Is how do we ensure that South Africa and the continent and broadly the poor countries in the global south are not last in the line to access any vaccine? So the first one is on they're all on ethical and social impact of the vaccine, on fake news, and the other one is now on the queue for the first vaccine.
So I'll answer
you can take that and then
I mean, I think that South African scientists and African scientists have to be there to support the clinical development of vaccines. And if we're not putting if our scientists are not involved, it becomes very hard to navigate the registration and access. So I think it's critical for African scientists and South African scientists to be involved in the clinical development of these vaccines and show our ability to conduct clinical research in Africa. African scientists and Africa is a very important continent for understanding disease and genetic responses to disease, and so we cannot be left out of any clinical development. And it's important that we continue to pursue clinical research in Africa. African scientists are highly capable. They're exportable across the world. We see a diaspora, they get stolen. And so the only way we can keep our capacity in Africa is to make sure that we pursue a research and scientific agenda that keeps our scientists here and responsive to the diseases that most afflict us in Africa. And so I think that we want to be involved in research, and we want to be part of the research endeavor, and we want to make a huge contribution, just like everybody else in the world. And we need to make science flourish in Africa.
Thank you, Glenda. I've got a minute now because technology gives you a hard cutoff. So
the medical team there behind South Africa's vaccine trial for COVID-19, giving updates on how they got to that point as that ended. Dr. Glenda Gray speaking about the importance of including African scientists and medical doctors in trials, in part because of our genes and genetics, and how perhaps a vaccine developed in Europe may not work as well